Something that doesn't really exist before is posing a threat to the entire world. The World Health Organization announced today that 3,000 cases have been confirmed in the western suburbs, and we are heading towards 10,000 cases. The new virus is spread from the infected cities Minneapolis, Chicago, Los Angeles, Boston, and Salt Lake to the neighbor country due to war travel.
Is named as MEV1. It stands for meningoencephalitis virus.
It is a paramyxovirus which infects the lungs and the brain. It causes both meningitis simultaneously, which is an infection of meninges and encephalitis. With an unusual incubation period, MEV1 kills an infected person in approximately 4 days after infection and within hours of onset of symptoms. Here is a model of the virus and how it attaches to its host. The blue is the virus, the gold is the human, the red is the viral attachment protein, while the green is the viral receptors in human cells. These receptors are found in the cells of both respiratory tract and CNS. The virus attaches to the cells like a key slipping into a lock.
From investigation, the Virology is shown to have maturity homology sequence as Nipah virus. This is a recombinant virus with some porcine rubella virus get inserted into the genome. I would say that this novel virus is a mutation.
It's hard to know what is it without knowing where it came from. World Health Organization had decided to send epidemiologists to Malaysia, the ground zero on Nipah. Negeri Sembilan, 2-hour drive north from Malaysia capital Kuala Lumpur, is an old mining town that had been hit by a killer that had never been seen anywhere of the world, Nipah virus. 257 cases of febrile encephalitis were reported to the Malaysian Ministry of Health, including 105 deaths. The virus was transmitted through the state of Peninsular Malaysia due to the significant movement of pigs. Active pig trade was normal practice in Peninsular Malaysia, which had a 2.4 million standing pig population at the time of the outbreak. As a result of trade, supposedly infected pigs were moved from farm to farm, both within and between states. With the discovery of the etiology of the pig disease, a total of 900,000 pigs from 896 farms were destroyed in the infected areas. Availability of ELISA had revealed that 5.6% of all examined pig farms were found to be positive for Nipah virus. Sungai Nipah, a village in the Malaysia Peninsula where pig farming is active, is where the first virus was isolated from a human case. This was the ground zero. Most survivors of Sungai Nipah have fled. 80 people from this village alone are died during the outbreak, and one family of every 3 had lost their loved one.
For now, it's believed that this virus is respiratory transmitted or even through the format. The format here refers to the transmission from the surfaces and in daily people touching people around and they're touching the elevator button, the doorknob or the table. All of this can become the format to transmit this virus.
How fast this virus multiplies depends on a variety of factors. For the first, the incubation period, how long a person is contagious. Sometimes people can be contagious without even having any symptoms. For the second, how big the population of people susceptible to the virus might be. Once the reproductive rate of virus is known, we can handle on the scale of epidemic.
The Nipah virus is normally carried by bat. The food bat transmits the virus to pig, which transmit the virus to human. Nipah virus has a lipid surface containing a shell of viral matrix protein. Inside the coat is long strand of RNA. The lipid surface contains two types of spike for fusion and attachment purpose. Once inside the body, the virus attaches itself to host cell, then release viral RNAs. Symptoms include fever, sore throat, muscle pain, pneumonia, and respiratory problems. Mortality rate is up to 75%.
Nipah virus can be cultured in many cell lines, including Viro, RK13, BHK, and porcine spin cells.
After successfully growing the virus using that cell line, CDC had tried to develop vaccine to halt the spread of NEV1. By using animal trial, they tried to use that viral combined with atropine to boost the immune response. Yet, no protective antibody produced, and out the virus killed all the hosts. After some trial, CDC had found that tested animals were able to produce antibodies and survive using live attenuated vaccine. Live attenuated vaccine contain live, weakened bacteria or virus that usually unable to cause disease. When it enters cells, it will still be considered as a foreign particles and trigger body immune response. The white blood cell will go through a trial and error period to determine the best way to kill it. When the white blood cell determine how to destroy it, they create memory cells to handle future attacks. As the body exposed to the same pathogen in the future, the memory cell react quickly without trial and error period, and this will allow body to destroy pathogen efficiently before it can cause illness. The vaccine was then formulated as intranasal vaccine and mass-produced.